So in this video, I'm going to be explaining how protein synthesis works. Now, a little disclaimer before I get started. If you have taken Bio 30, I'm going to be explaining protein synthesis a little bit different than I did in Bio 30 because in Science 30, it is not required for you to know as much about the process. And unfortunately, I have to actually kind of teach it incorrectly in comparison to what we know in Bio 30. So if you've taken Bio 30, you kind of have to just ignore what you previously know about this process and just listen to me today. So, we're going to talk a little bit about what proteins are made up of. Remember, proteins are super important because they make up things like hormones and enzymes, and like our ligaments and tendons, and things like hemoglobin that transports oxygen in our blood, and antibodies, and so many other really important things. So we're talking about what they're made up of, and they're made up of amino acids, they're called. So amino acids are the smallest building blocks of them, and there's 20 different amino acids that can make up our proteins. So we can get some of these amino acids just from our body. Some of them we have to get from our foods. So that's why it's super important that you have a diet that includes protein, whether that's a meat byproduct or whether that's actually like some other form of protein like legumes or something like that. It doesn't matter what you're eating as long as you're getting some protein somewhere. So basically, depending on those 20 amino acids, how they're sequenced in a long chain of amino acids makes up a certain protein. So, like, for example, if I'm making a specific enzyme that breaks down a certain thing, it has to have a certain combination of those 20 amino acids. And we call them polypeptide chains when they're kind of connected together. And then we look at our DNA, the sequence of the DNA, those letters that we have in our DNA, and we use them to basically read them as instructions on how we create this amino acid chain. So when we look at a gene in our DNA, that normally we talk about genes as like the eye color gene. Well, in reality, a gene is actually coding for a protein. So for example, if we're talking about eye color, the protein that's being created is the protein that makes the color in your iris. So it's a little bit kind of a weird kind of mind shift that you have to think about proteins when we talk about them, because we talk about DNA like passing on traits a lot, and I think everyone gets that, but I don't think anyone really understands what proteins are because it's like the kind of the behind the scenes of what your traits are showing. And then just a reminder that polypeptides are just a long chain of amino acids, and they're linked together with something called a peptide bond. So in your guys' data book on page 13, you have a table that looks like this. We're going to be looking at that table all day today. And this is a table that you have to refer to in order to find the amino acids. You don't have to memorize this table. You can always use it to look stuff up. So amino acids are coded from 3 nitrogen bases. So you can see all of the different options of the 3 nitrogen bases and the different amino acids. Each 3 nitrogen base is called a triplet of DNA. So you can see the title of our table is called DNA-based triplets. Okay, again, if you've taken Bio 30, this is a little bit different of a table than what I give you in Bio 30. Notice that it's DNA-based triplets instead of a different thing that we were doing in Bio 30. So just kind of to point that out. If you haven't taken Bio 30, just ignore me. So for example, if I was giving you AAA as my DNA triplet, I would say that this is lysine because I have A, and A is lysine in my table. So I'm using this data table to figure out what my triplets of DNA code for. Which of the 20 amino acids do they code for? What is it, lysine, or is it histine, or proline, or isoleucine, or valine? Like which one in the table is it actually showing? And we're always using that page 13 of our data book. So I'll also connect a copy of this table on Classroom so you guys have access to it. So it's a little bit easier for you guys to look stuff up, because I realized if you don't have your data book anymore, maybe you didn't bring it home with you because we haven't been using it in a while. It's not in your notes, so I will link it on Classroom. So just a couple of practice questions just to kind of get you used to using this table. So you can kind of pause me and find it yourself and then press play when you want the answer. I'll just kind of show you kind of the baseline of how you use this table. So the first base is a T, and then there's a G as the second base, and the third base is a T. So this one should be cysteine. Second answer is C, A, T. So that one should be histidine, and ATG, so A and T and G. So this one is a little bit of a different amino acid because it's either methionine or it starts. So it will come at the very beginning of each gene that I give you. So if there's like a sequence of DNA that is a gene that codes for a specific protein, it should start in the start DNA triplet. And then the last one is stop, because we have TGA. So there's 3 different stop options. Again, like the start one, that one will come at the very end of a gene so that the whole process knows that it needs to stop at that and not copy any more letters. So those were all of the questions in your book. I just wanted to go through and make sure you understand kind of how this process works a little bit more. So you can either write this down somewhere, or you can just kind of follow along with me and just watch what I'm doing. So normally there's 2 strands in DNA. We have 2 sides to our DNA. So like in this picture, we have the pink side and the blue side or purple side. So you have a non-coding strand and a coding strand. So for example, if I gave you a non-coding strand of DNA, and I asked you to make the coding strand of DNA, the coding strand is always the one that you want to use to make your amino acids from, or the template strand, sometimes it's called, or the sense strand. Those are my 3 options of words I could use. So I'm literally just matching up the letters. If there's a T, I put an A. If there's an A, I put a T. If there's a C, I put a G. And if there's a G, I put a C. So I just kind of copied down the letters to make the coding strand. This is kind of the process that you would have gone through in DNA replication to make a new strand of DNA, and that we practiced a couple days ago. So when I'm making the amino acids from ATG, etc., I'm just looking them up in the table. So ATG is right here. It's start, because it's at the very beginning of my little gene. Obviously, genes would be way longer than just 4 triplets, but I just didn't have a lot of space on my page to show you. AAT. So we have AAT as is asparagine. And then triple T is phenylalanine, so that's why I put phenylalanine, and then TAA is a stop. So those are the amino acids that go with the coding strand. Make sure you're always using the coding strand, because if you use the non-coding strand, you would get 4 different amino acids, and you would get the question wrong. So always use the coding strand of DNA. If they just give you a single strand of DNA and just say make the amino acids, and they don't tell you if it's the coding or the non-coding, just assume that it's the coding strand, so you don't actually have to take it and make the complementary strand of DNA. You just make it into amino acids. If it doesn't mention that it's the coding or non-coding, just assume it's the coding. So here's another strand of DNA. I have the non-coding strand, so I have to make the coding strand first. So it would be ATG, GGG, CTA, TAG. So that's just making sure that all of my nitrogen bases pair up with the right letter. ATG is start. GGG is glycine. CTA is leucine, and TAG is stop. So it should be start, glycine, leucine, and stop. Start, glycine, leucine, and stop. Perfect. So that is how we would do these questions. This is kind of how I expect you to, or what you should know kind of from this video, is being able to do like matching up the letters and then just looking them up in your data table. And as long as you have the data table in front of you, it shouldn't be too hard to do this. So just to make sure we understand the process of what is called protein synthesis, also known as just like creating proteins, so taking DNA and making proteins. I would take my DNA strand, which have letters. Those are my nitrogen bases. I would take the coding strand, let's just say this is the coding strand, and then I would make them into amino acids. So the first one, I know it's kind of hard to see, it's a grainy picture, TCA. I looked up on my last slide, is serine. And then the next is CTT, which is a leucine. And the next is GGT, which would be glycine. Eventually, when you copy all of the amino acids into copy all of the DNA into your amino acids, you'll eventually get a long strand of amino acids. And later, after we're done this process of protein synthesis, this isn't a protein technically, a long chain of amino acids, it's just a polypeptide chain. And it would, like, as it's traveling through our body, it would get like tangled around stuff and just kind of be a nuisance. So like it wouldn't only be 3 amino acids long, it would be like potentially like hundreds of amino acids long. So we want to just kind of package it into like a little nugget of DNA, a little nugget of amino acids. So then it's actually like a little bit easier to move around our body. So this is my polypeptide chain. This is my protein. And every single protein, when it has the specific combination of amino acids and you package it into the little nugget, it has a certain 3D shape. Just like when we were talking about organic molecules, and every organic molecule has a certain 3D shape that we were drawing on the board, similar to that. And that's how it can interact with other things. So, like if it's an enzyme, for example, its 3D structure would match with a certain molecule and then break that certain molecule down. Okay, if it doesn't have that right 3D structure because it has the wrong kinds of bonds, or maybe it has the wrong amino acids in it, it's not able to kind of go into its 3D structure correctly, that would mean that protein is basically useless and we couldn't use it in our body. So we wouldn't be able to break down that certain structure that we're trying to break down. So that's why we have to kind of package it at the end. So we're going from DNA, we're taking the coding strand of DNA, we're using our table to make the amino acids, and then eventually we would kind of just package that DNA into, package the protein into a little nugget. I keep saying DNA. So that's the process that you guys have to know. Basically just knowing how to use that, the chart with the DNA triplets and being able to look up what one is what. So always making sure that you're using your coding strand of DNA. And that's basically all I have to teach you about protein synthesis. So hopefully that makes sense. I'll talk to you guys later. If you have any questions, let me know.